And I'm deliberately being complex here because I want you to see that um, in all of these pathways, it's not a linear pathway, and that there are a number of different branch points that that the different molecules can make to decide to go into a pathway or not. And that's the trickiest thing for us. Also, these red circles are indicating we already use drugs that are targeting cancer metabolism. So we know that this is something that can work in the right cancer, in the right setting. But I have talked about imagining cancer metabolism as looking a little bit like the London Underground tube map. And last night I found one. So some geek has made a beautiful map of the London Underground and actually turned it into metabolic pathways. What I like to describe is during therapy, it's like blocking off one of these tube stations, and a molecule that's trying to be made via this pathway, if we put a therapy here that blocks that pathway, well, actually, people often ask me, why haven't we found a cure for cancer yet? Well, we've found many, but cancers are incredibly clever and they will find a way around whatever therapy pressure that you put on them. And this is called resistance, and this is what causes a cancer relapse. And it's not always, there's lots of reasons that it occurs, but what the cancer will then do in the case of cancer metabolism is it will go, I can't go that way anymore. I'm going to get to that same place by taking this different line. And what we try to do then overall in our research is understand when we put therapy pressure onto these cancer cells, how do they reprogram their metabolism? And is that predictable? Are they always going, oh, can't go that way, going to go this way? And if they do, then have we got a drug already? Or can we design a drug against another stop in the tube map to prevent the cells from being able to create that macromolecule and beat them there and starve the cell of their energy that they need? So in the search for new therapies to target metabolism, many medicines have been first discovered in plants or other biota, and some colleagues of ours isolated a molecule from a sea squirt that they found in the Great Barrier Reef. That sounds very random, it's not very random. But the short story is this molecule is toxic when you, is everybody still hearing me? When you add the cells, when you added the molecule to cancer cells, but it didn't occur, it wasn't toxic when you added them to normal cells. Well, this is just brilliant news as far as we're concerned, because we don't want to be toxic to normal cells. We just want to be toxic to the cells that use these unique metabolic pathways. And we know from the work that we've done so far that these metabolic pathways work on this glycolysis pathway. So the aims of the project would be to identify the mechanism of action and understand exactly what it's being used in. But this would help us because firstly, it could identify what's an important tube stop for cancer metabolism. And it also might be able to provide us some important structural information about what might make a really good drug. And so what we would be doing in the lab, I need to hide you all again, one of two experiments. So in both cases, we grow prostate cancer cells in a plate. And in the first experiment, we would treat the cells with our compound and with a couple of other inhibitors of different tube stops. And we would monitor the way that the cells grow. So this is a machine that takes photos of the cells every 2 hours, and we can have a look over time and we can see how that changes the cell growth. And at the end of that time, we can use different dyes to have a look at the way that the cells have changed their metabolism. So we can look at the activity of the mitochondria. We can look at the nucleus and the shape of the nucleus. We can look at the amount of lipids that are being stored. We can assess the amount of glucose that the cell is taking up. And then we use this machine, the InCell, which can take a gazillion photos. It can take about 10 photos of every well in the 96 well plate in about half an hour, and I have spent days taking like 20 photos previously, so this is an incredible piece of equipment. And then, of course, we've got a lot of image data which we can use segmenting packages to analyse, and this can give us our digital data, and then using packages such as R or this cell profiler package, we can end up with numbers that tell us where there are changes in things like this is an example of lipid content. The second experiment would be putting the same cells, into a different type of plate that we use for a very specific machine. We would use the same treatments in this, but now we would be measuring the way that the cells actually respire. So are they using, so this is a little probe that comes down and creates a tiny little pocket of media where we can make very sensitive measurements of whether the cells are using the mitochondria or glycolysis to their energy production. And when we add our different inhibitors in, we're very in understanding how it copes under that new stress. And so we would use the same combination of blocks or inhibitors of different tube stops to understand what the response was and see whether we can kill these cancer cells in a dish. Okay, so finally, advice. I shouldn't laugh. So actually, I do have some advice. My first piece of advice would be there are many pathways to a career in science. From the time that you sit your HSC exam, to your final exams, to the time that you come into a degree and think, oh, this is not quite what I thought it was going to be, to the time where you start your career in pathology and just think, I want more than this, to the time that you go backpacking and accidentally find yourself with a job. Don't ever think that I didn't quite get the mark I wanted, or I didn't quite come in, I'm not quite doing the right subjects, is that roadblock is a dead end. It just never is. And you can be a little bit like a metabolizing cancer cell, and there are other ways to get there. So never feel like you've reached the dead end for science careers. The other thing I would say is open yourself up to opportunities. I am always amazed at how often I think, God, I was just in the right place at the right time. But somehow you were in the right place at the right time. You know, you put yourself out there and you have spoken to somebody, or you have attended a seminar of a person you admire and had the chance to talk to them afterwards. You know, you can create a little bit of that opportunity as well. And while life is full of serendipity and random chance, some of that isn't as random as we might like to think it is. And finally, the last thing I would say is science is a really marvellous way to build resilience. There are many more fails than there are successes in science. I can't be the only scientist that told you guys that. And it's a really critical skill. So certainly I spend about a day after somebody tells me that my grant didn't get up, or my paper is going to be rejected, to just back myself and tell myself that I haven't got this far by being rubbish, and that they're just words, and dust myself off and do it all again. The successes, though, are just ridiculously sweet by comparison, and it's a really rewarding career. So that's all I was going to say. I hope I haven't taken up too much time, but I do hope we have a little bit of time for some questions. And I'm going to ask Diana and Emma to help me to manage those so that I don't miss anybody.
Yeah, sure. Let me just have a look though.
Hi, I've got a question from Matt Chow. So he's asked, is it true that cancer survivors are more likely to develop it again? So I've got two answers to that. Sometimes you can have a cancer that can be really easily managed, and you would anticipate to never see that cancer again. Prostate cancer actually, and often that's related to how early you detected it. So prostate cancer is a very good example of that. If you can find it before it's spread outside the prostate, the treatment is to remove the prostate, and we've got an effective cure rate of about 95% in men who do that. However, if we do not have a very good cure if the cancer has metastasized, and that is that it's spread to another part of the body, and that is true of almost all cancers. The success of the new immunotherapies is outstanding, and I can't wait to see how well that does. And so I might be giving you outdated information, but we've not collected the data to prove me wrong yet. But what we try to think about for things like breast and prostate cancer is how do we treat this not as a disease that is going to kill you, but is a disease that you'll manage. And that's why we're constantly looking for, okay, what's the next resistance pathway? What's the next resistance pathway? So, and I've given you a very metabolic centric view of that, but people look at many, many different cellular pathways to do with metastasis. How could we stop the metastasis from occurring? How can we regulate the immune recognition of these cells? So your body would be destroying precancerous cells all the time. And it's when that cancer cell starts to hide from your immune system that we end up with a disease, which is rare. Okay, that's rare. And so once you have a metastatic disease, then it does become a series of what's the next drug and what's the next drug and what's the next drug. Yes. And what we hope is that we can find enough of those that you are managing a chronic disease and not dying of that disease. Yeah, so I think that answers Brianna's question about analysing. Yep, we look at, so I guess there's metabolism and there's metastasis. And metabolism is the way that the cell is using the energy, and metastasis is the way that the cell is escaping from the primary tumours and going off and making tumours in other parts of the body. No, we don't always consider metabolism when we're looking at specific drug inhibitors, and most drugs are either targeting the way an enzyme folds to be able to be able to function, and not always in metabolic pathways. Most often in what we call cell signaling pathways, which are kinases or other key molecules. So first of all, we weren't looking at metabolism. We've only started really looking at metabolism again in maybe the last 10 years. And we got very hung up on looking at kinases and the way that cells signal, because that's what we could find first. We looked at proteins, we've looked at DNA and RNA, and now we're back at metabolism, which is where we started. So it's been, technology drives a lot of our discoveries, and it drives our ability to develop new therapeutics. And then we all have to get in a room and chat together and use a multitude of those. So a metabolic therapy on its own is probably not going to work. We need to talk to the people who are developing kinase inhibitors and developing immune checkpoint inhibitors and developing metastatic inhibitors, use those as an arsenal. Yes, absolutely. So the next question, I just have to see who that's from. From Willow, have you started tissue engineering cancerous tumours to watch the way they react to treatment without testing on a human patient, and what opportunities are within that field currently? So yeah, typically and historically, yeah, we absolutely cannot go straight into humans. There's a lot of around that actually. We look in the dish first. That's the cheapest and easiest way, but it's but it lacks this complexity of the 3D system, and then the 3D system with blood vessels, and the 3D system with all your other tissues. So the next thing that has to occur usually is that it goes into an animal model, and there are usually a number of different models that we can use to recreate cancer, typically in mice. Then there is a huge field that's trying to create all of that complexity in 3D models in the dish so that we don't have to use mice. Yes, so absolutely, we don't put things straight into patients. Was there a specific career that you had in mind to pursue? I did think pathology. I nearly changed to a forensic science degree because that was just so cool in the late 90s. But I'm glad I didn't, because I think the cell biology, I could have gone into forensic science if I'd really wanted to, but actually the opportunities in a applied chemistry degree were probably not the ones that I was interested in as much as a cell biology, immunology, microbiology sort of degree. I definitely hadn't anticipated anything that happened to me overseas. Was maybe going to come back and sit GAMSAT or something like that. But yeah, that is part of the life journey, that some things happen that you don't anticipate, and actually you can turn back and be glad that that you said yes to an opportunity that came up, and that's why I gave that advice. Yeah, so from Nick, does treatment working on primary tumours, or there's a possibility of it working on secondary tumours as well? I'll answer your first question first. Yeah, we actually work only on the secondary tumours. So as I said, for men with prostate cancer, their primary treatment is excellent. And if they catch it in time, then we never see them again. But we are mostly working on the changes that occur for the men with really advanced disease, and we absolutely study the way that metabolism changes when the cells become metastatic. A lot of things change in that context, and not just their metabolism, but their metabolism does always change. So it does give us an opportunity to capitalise on that difference, and to identify potentially a very easy drug and pop that into the treatment regime as well, and potentially have some really good effects. So things like drugs that we've used for diabetes for 30 years are now being tested in the context of cancer with some really promising results. And we call this drug repurposing. And that is a very quick clinical turnaround. We can put those into patients really quick. We don't have to go into mouse models for that. You had a second question, Nick, which was, is there a possibility of part of the cell affected by the mutagen and destroying or treating that instead of destroying the entire cell, to avoid destroying cells in the case where it can be very detrimental? Yeah, that would less so. Once a cell is cancerous, there's really no going back for that. But yeah, what we want to avoid is, if I'm targeting cells that are highly proliferative, it's the same as chemotherapy. So hair follicle cells would be affected, and the lining in your lungs is affected, because these are cells that have a rapid turnover anyway and have quite a similar, not the same, but a very similar metabolic pathway use as a cancer cell. That's what I think might be so exciting about the immune checkpoint therapies. So these are the ones, effectively your cancer cells themselves from the immune system. The immune system can't see them, and this basically says, this uncloaks them, this therapy. And if we are able to reawaken the immune system to recognize what's a tumor cell, the immune system is incredibly good at finding and destroying those cells. It's not a problem to destroy a cancer cell. We can manage to do without those. You're right, it does become a problem when these cells, when the cells that are otherwise have a rapid turnover are also affected. And yes, we would try and do anything we could to prevent those side effects. And one from Brianna, does the immunotherapy research or editing specific genomic sequences ever overlap your own research on drug inhibitors? That's a tricky one to ask. So not so much at the drug inhibitors, but in trying to identify what would make a good target of the immunotherapies. So this is where we would identify a sequence that is prostate cancer specific and get the immune system to react, see, and treat it as foreign. Really just in combination. Yep. I'm not looking. So there are other kinase inhibitors, for example, BRAF inhibitors that only recognise the mutants. That's not really what I'm looking at. I'm not finding a lot of mutations in metabolism, but they are there and people are studying them. And if you can find that, then you're onto a winner. It can be very specific for really more for the rare cancers like rare brain tumours, etc. Very exciting stuff. So I did find a job while backpacking. I would say that if you take a degree, travelling with you, it can be very easy to find a job because you can use that specialisation to access different visas and to and get a little bit of extra money than you do as when you want to go backpacking. I just used an agency, a job agency. This might be slightly off topic from Liliana in regards to enzymes. Do you mean in terms of inhibitors for the lock and key or induced fit? I mean, is that what you mean, Liliana? Can I get some feedback from you for a sec?
She said yes. That's what she
Okay. Yeah. Usually there's we do what's called a screen, and we will take all comers. We really don't have a preference, as long as it can block the pathway effectively and specifically. And so, what we'll do is use those 96 well plates again, actually usually 384 well plates these days or even more, and be screening 40,000 compounds, and whichever one has managed to kill the cells, we're on to that one. So I think we've got time for one more question. So thank you very much, Diana and Emma, for letting me go on for so much longer than we planned. And thank you all very much for coming. The last question was, the process that causes the immune system to properly recognize the cancerous cells. Actually, what the drugs do is the cancer cells have a way to tell the immune system, the immune cells, so we've got immune cells going around through our body all the time, sort of checking out, making sure everything's okay, treat them like a policeman doing on the beat, making sure everybody's behaving themselves. When they find someone that's misbehaving, a cancer cell that's misbehaving, the cancer cells actually are, I said cloaking, but they're really just telling the immune system, you don't need to react to me. And it's activating a pathway in the immune cell that says, oh, I don't need to react to this. So what the therapies are doing is blocking the pathway that tells the immune system that it doesn't have to react. So it is a bit of a long way round, but then the immune system doesn't use that pathway, and it actually does now react to the cancer cell that it's found, and it activates that way. So I think we have Yes, if you have any further questions, you can absolutely contact me via the STEM Schools email. They will be sending out my slides. I don't know. Hope you find those interesting. And certainly, yeah, I'm happy to answer any other questions that you have about a career in research, the steps to get here, what day to day looks like for me that we haven't had time to go through today. Oh, you're all marvellous. Thank you for coming and thanks for taking an interest. I find it a highly satisfying career. I think the best thing about it is I learn something new every day. And some days that's the worst thing about it because I have to learn something new every day. But also, it is when you get those wins, it's just an incredible feeling. And I work with some really amazing brains, which is always really inspiring. So, good luck, everybody, on your science journey. I really hope I meet some of you in the future, potentially at QUT. And thanks for listening.
Thank you, Jenny.
Bye, everyone.